come over.
I want to start you with some food for thought, because what I'm going to talk about today are some studies that are going to challenge the notion that sex is all about reproduction. Obviously it is, but it's about more than just that. So let's take some food for thought. We can say that we've evolved a nervous system that acts in the interest of our gonads, and one that's attuned to the demands of reproductive competition. And I think everybody in this room and probably the world over would say, well, yeah, of course, that's true, because if we don't reproduce, then the species is gone. So we can look at that as an ultimate goal for reproduction, but there's other ideas that have been out there, and out there for a very long time. This one from Richard von Krafft-Ebing, back in 1886, which predates Pavlov, where he says erotic fetishism makes an idol of physical or mental qualities of a person, or even merely of objects, like your vacuum cleaner, because they awaken mighty associations with sexual pleasure. So sex can exist for reproduction, and it can exist for pleasure. So either every time we have sex, we're trying to reproduce, or every time we have sex, we're trying to feel good. And reproduction becomes then icing on the cake. So what is it? To understand that, we have to really ask the question, what do we need to learn about sex? Isn't it all just innate? Or are humans the ones that learn about it and animals just do it, right? So of course, as you look here, I don't even know why the bunny rabbit would try to choke the chicken. That's probably the last time Sparky ever did that. And I don't even know what to say about Bob there. But if we think about it, we've been studying two different things in animals and in humans. In animals, we've been studying these more reflexive things: autonomic activation, genital reflexes, sensations, innate copulatory behaviors. And these things involve lower, quote-unquote, parts of the brain, like the hypothalamus and the spinal cord and the brainstem. And we say, well, that's where their sex is. They just do it. They do it as a function of their hormones. They do it as a function of, say, reproductive cycles that may happen, say, during springtime, but not during fall or winter, when resources are available, blah, blah. In humans, we talk more about an incentive system, where there are Pavlovian associations between stimuli and rewards or punishments that come after them, and operant associations between what you do and the rewards or punishments that are achieved by that. And you can imagine sex, think of sex in those terms. And these involve limbic and cortical structures that you can certainly see with an MRI or see with a PET scanner, but you can't really see well these other regions. They get activated, but you don't know what that means. So what's what? Well, we started to think about this deeply and understood that the Masters and Johnson, EPOR model of human sexual function here, but it's really about arousal, with desire coming before arousal, reaching some plateau while you're having sex, reaching orgasm, and then producing an inhibition or refractory period, that really you have expectation in this time, which is wanting sex. You have consummation of sex, liking it, hopefully, while you're doing it. Hopefully it's good, then you like it. And then things that you learn as a result of what you've done. I mean, you're learning things throughout, but the result of what you've done then feeds forward on your expectations for next time. So we ask the question, do animals do anything like that? I mean, what would be the role of learning in sexual responding? Well, animals have to learn their own patterns of arousal and desire. They need to learn where potential sex partners or mates are. They need to overcome obstacles to sex or mating. It's not just we who have to do this in bars. They have to do this in the real world. They have to recognize which ones are fecund and which ones aren't. They have to show courtship behaviors that work as opposed to those that don't. They have to show copulatory behavior that works as opposed to those that don't work very well. And they show sexual partner preferences, despite the fact that most animals are described as promiscuous rather than monogamous. So we have taken sexually conditioned place preference. This is a psychological sort of paradigm where you have two goal boxes here. One's dark, one's light. Animals, rats are nocturnal, so they tend to prefer to be in here. So what we do is we bait the light side with something good. It could be heroin, it could be cocaine, it could be the feeling you have after sex. We bait the other side with nothing, and then on a final test, put the animals in the middle, open up the door, and see which side they stay longest on. And it turns out, if they like it, they're going to stay longest here, thus shifting the preference from an innate preference here to now acquired or learned preference on the other side because they're expecting the reward state. Everybody make sense, right? Okay, so one thing we know about sexually conditioned place preference in males is that ejaculation is necessary for the conditioning. If they don't experience the state that they're in after ejaculating, you don't get a preference. We also know that you get a stronger conditioned place preference when the males are more aroused prior to ejaculation. So for those of you who have done anything naughty in your lives, you know how naughty can really embellish the impact of orgasm. We know that the acquisition is blocked by the opioid antagonist drug Naloxone, but it's not blocked by dopamine antagonists, which is a very important idea. Dopamine antagonists and dopamine itself are really activating the expectation of the reward, but not the reward itself. The reward is an opioid-mediated event. What about female rats? Well, it turns out for females, it's not an ejaculation. They're not toys of the male. They really like the ability to control, to pace the initiation and rate of copulation. If they can control the clitoral stimulation that they're getting in time, which is actually reproductively salient for them, they will really, really like to have sex. Not only that, in this divider paradigm here, there are 4 holes down here. We can make it with 4 holes or up here 1 hole. The problem with 1 hole is that he gets his head stuck in it. So she really can't do what she wants to do when she wants it. 4 holes, what she can do is aim over here, he'll put his head over here, then she'll pop through one of the other ones. So she has complete control over what she gets in that paradigm. Well, it turns out they love that, and they will show a place preference for 4 hole over 1 hole. Timed clitoral stimulation, which we can do with a paintbrush, does exactly the same thing. And it's blocked by naloxone, but not by dopamine antagonists, which means, we talk about sex differences, vive la différence, but it turns out the brain is the brain. Men are not from Mars, women are not from Venus, we're both from Earth, and we have a brain that grew on Earth. So we have studied now conditioned partner preference as well, using the same logic as the place preference studies. So in males, for example, we gave males copulatory experience with almond-scented receptive females. That was very simple. We put the almond on the back of the neck in their anogenital areas. So that would be a paired group. So we're pairing now the odor with that post-ejaculatory reward state that the male will be in. Or in an unpaired group, they get unscented receptives, so they don't know what the odor means. You can make the odor mean nothing by giving it to them in their home cage, but giving them copulation with unscented receptives. So now they've had the same experience with the odor in terms of time, but not in terms of meaning. Then what we do is we give them an open field test. We wanted to call it a ménage à trois, which most people know what that means, but the editor of the journal didn't like that. Being very English, he said, that's a French term. People won't understand it. So he said, instead, use triad copulation, which is very boring. But the male gets a choice of 2 females, but both of whom are receptive. One is scented, the other's unscented. And we can ask the question, who do you prefer? Do you have a preference? You're supposed to be promiscuous. So maybe if you've been in this group, you're going to prefer this one because she's novel. If you've been in this group, you're going to prefer this one because she's novel. And then you're going to be spreading your genes far and wide in the pool because that's what all men want to do, right? Okay. So what happens? Here's training. The odor is put on the back of the neck in the anogenital area. So here's the female approaching the male. She hops over him so he can sniff and lick her vagina. Then she kicks him in the face and runs away. It's very grade 5. So you can see the male chasing her, and finally she holds a lordosis and he can intromit. And he'll do this repeatedly till he ejaculates. That's training. This is testing. So here's the pair trained male. 2 females are put in. And he goes and he chases and then copulates. So who does he choose? Well, it turns out when we look at who he chooses for first mount, first intromission, which is the vaginal penetration, or first ejaculation, and then the number of mounts, intromissions, and ejaculations, what you see is that in the paired trained group, the males are ejaculating preferentially with the familiar female. And in fact, they're ejaculating more with the familiar relative to the unfamiliar female, whereas they're mounting and intromitting with whoever's in front of them. So being a good male, they're having sex with whoever's in front of them, but they're mating with the familiar female, which means they're going against promiscuity and showing elements of monogamy because of Pavlovian conditioning, not because they're pre-wired for this. The males in the unpaired group, well, they don't show a first ejaculation preference, but they still show a preference to distribute their ejaculations to their familiar female, which is the unscented, relative to the scented. So they too are able to show this pattern. And it turns out that ejaculation, like with place preference, is necessary for this, because if we give them intermissions but no ejaculation, or an ejaculation but we pull the female out, or we let the female stay during that post-ejaculatory interval with the male, that's where the conditioning occurs. And this is despite the fact that when we look at the males, they're asleep. Dude's asleep, but he's processing because she's still there. So we can affect this with the hormone oxytocin. We give this to them on their first conditioning trial, and then we look 4 days later, when there's no oxytocin left, we ask the question, can we enhance or embellish this conditioning? And it turns out, indeed, with oxytocin and not vasopressin, we can enhance that distribution of ejaculations. So what does that mean? Well, it reminds me of Stendhal's principle from De l'Amour in 1822, when he said, if the couple remain in close facial contact after the generative act, which at that time meant sex, he shall come to associate the flower of desire with her features that to him shall be salient, and he shall seek out similar features in another, should this bond be broken. In other words, beauty is the promise of happiness. What does that mean? It means he has a type, and the type is conditioned by reward, by pleasure, by his experience. And he calls that process in 1822 crystallization, the operation of the mind that draws from all that presents itself, the discovery that the loved object, i.e. her face, her hair, her cheekbone structure, whatever it might be, has some new perfections that he's now associating with that state. What it means is, when you're cuddling, you're activating these processes. Well, what's cool about these is we're not just looking at copulation now. The odor can actually be a priming cue. So we can say, what's the meaning of the odor? What is it to a rat? What is the meaning and what are the mechanisms of that meaning? And we can present the odor alone as a prime. Just like we would see in a magnet if we're looking at porn. It's a prime. It's not real. It's just a priming cue to get us going. So we can ask the question. Well, it turns out when we look at the conditioned versus unconditioned odor, the conditioned odor activates FOS, which is a nuclear protein in all cells that are activated, in regions of the hypothalamus, like the medial preoptic area, but also regions that are associated with reward processing, like the nucleus accumbens and the amygdala, and the ventral tegmental area, the septum, the insula, anterior cingulate, and piriform cortex. These are all regions that we have seen activated in humans when they're seeing sexual circumstances or erotic circumstances that they find arousing. We also see that it activates dopamine release in the nucleus accumbens relative to the unconditioned. It activates oxytocin and vasopressin neurons in the paraventricular and supraoptic nuclei where those neurons reside, and it activates GnRH neurons and increases plasma testosterone levels, which means that if you're getting something you like, that you prefer, that you're attracted to, you're actually more able to reproduce with that individual rather than being less able, because you've got this activation conditionally of these systems. And this, I just said this, so I don't have to show it to you. What about females? Well, again, I told you that for place preference, they like the 4-hole over the 1-hole. So we asked the same question. Would that influence in females their choice of male? Right? Now remember, females are supposed to be more choosy. Darwin said that. And why? Because you possess all these resources that you're going to give to this offspring for 9 months for us, for 21 days for a rat, and they only live 2 years. But imagine that your choice really has to be the good one. Yeah, we can go and spread our seed all over the place, but you have to make that real big choice. So it better be for the reproductively salient male. Okay, so we're letting her have sex with a scented male or an unscented male. right? So the odor versus with pacing, the ability to do that. Unpaired is the odor and no pacing. How do we do that? Real easy. We remove the barrier. They don't know any different. And of course, random means they get sometimes the barrier and sometimes not. Then we give them a ménage à trois with an unscented and scented male. Notice they're tethered because the males are, female rats live in a matriarchy that's run by the females. So the females control everything. And the males are very cooperative. So if this one's scented and this one's unscented, they're going to crawl all over each other. And eventually they'll both be scented. So we had to keep them tethered to one side or the other so she can choose. Well, what does she choose to do? In the paired group versus the unpaired or random group, she solicits first the scented male. She solicits more the scented male. She takes that male's ejaculations preferentially, and she shows higher lordosis magnitudes with that male. What does that mean? That means she's much more likely to get pregnant by the male that did her better, not necessarily the male that is more reproductively relevant. But the reproductive relevance here is that he's the one that gave her pleasure. She doesn't know the other one, and why should she care? And it turns out it activates FOS. These black dots are FOS. It activates them in a multitude of brain areas that are associated with copulation and also with bonding. But it's not just this. Female rats who are allegedly promiscuous, unlike the prairie vole who are allegedly monogamous, they can actually show mate guarding. They don't even need the odor. As long as you give them copulation with a one and only one male, so their first sexual experience and subsequent experiences with one and only one male, so that's the boyfriend, she's the girlfriend, and the hembra otra, or the hoe, is the competitor. So here you have two females and one male, but one of those females, or to her, he's her boyfriend, and she's a competitor. What is she going to do? Well, here's what she does. She hovers and presents around her boyfriend. If the competitor comes down, the girlfriend actually gets in between them, so she blocks them. And if the competitor runs around and solicits the male, the girlfriend's actually going to mount the competitor, which is an aggressive response, pushing her head in the corner. So she will aggress against her. And eventually, she just has to do this once, and the competitor stays away. It's kind of like those glances. I don't know if you've been at bars. You watch, I've got my boyfriend here. I see another woman. He's looking at this other woman. I'm going to look at her and tell her I'm boss. And we see this happen, and that's mate guarding. Okay, well, rats do it their way. We do it our way. Well, it turns out that it activates FOS, the boyfriend, relative to a different male. So paired versus unpaired, activates FOS selectively in regions of the brain that are associated with bonding and with learning. Not only that, it activates oxytocin and vasopressin neurons. So we look at double labeling. So the black dot is the FOS, the cytoplasmic label is oxytocin and vasopressin. You can see in the supraoptic and paraventricular nuclei, oxytocin is activated preferentially in the paired relative to the unpaired groups. And vasopressin is activated preferentially in the SON, which is a region that projects to the posterior pituitary, releases vasopressin that will increase your heart rate. So you see the one you love and your heart jumps out of its body. Why does that happen? Because you're getting a massive release of vasopressin. Not only that, but we can potentiate with oxytocin. We potentiate with oxytocin solicitations and that presenting behavior, and as a consequence, they get more intromissions by the male. So just like what we saw with the male, we see it with the female. But unlike the male, vasopressin activates this blocking behavior selectively. So it's a bit of a double dissociation. We can knock it out with a lysine-specific demethylase inhibitor. Now, what is that? Well, genes, in order to be expressed, have to, they're all packed up, right, along these histones. And you got to unpack them in order to be able to express them. And part of the enzymatic relationship that makes that happen is they get demethylated. So the methylation kind of keeps them all packed up. The demethylase unpacks them, allowing them to be expressed. If you inhibit that unpacking, then you inhibit the expression. So we put the demethylation inhibitor into the brain, and lo and behold, we knocked out the initiation of the hovering behavior. We knocked out the initiation of the blocking behavior, which means that their first experiences are changing their brain epigenetically and turning them from promiscuous to monogamous. Not only that, we deactivate now with the demethylation inhibitor the release of oxytocin in both the SON and the PVN. So we don't get the oxytocin response, which means that in order to get it, you got to demethylate it, which means that their first experiences are activating the oxytocin gene expression. We can also block it with naloxone, which is that opioid antagonist. So it's meeting all the same criteria that we saw with place preference. So here you can see that animals that are given naloxone on the first 9 conditioning trials, and it only has a half-life of 5 hours, so it's out of the body 4 days later when we do the final test. So it's all about expectancy. Well, if you expect sex without reward, you're not going to solicit very much. You're also not going to lower dose very much. And as a consequence, you're going to get fewer intromissions and fewer ejaculations because you're not into it. And male rats know when the female's not into it. Naloxone also blocks that conditioned ejaculatory preference in males. So the ones who get naloxone during training, and again, there's no naloxone on the final test, they actually shift their preference from the familiar to the novel. The ones who got saline are now showing the preference for the familiar and the ejaculation preference that you see here. So blocking reward blocks the activation of all of those phenomena, oxytocin, dopamine, the gene expression that's required for that. So pathways for sexual reward and incentive responding overlap in the brain, as Larry Young has shown, with pathways for bonding. This has important implications for drug addiction as well as for anything that we might study that involves feeling good about it. And we can put together a circuit diagram, which is what this is, but at the center of the circuit diagram is this medial preoptic area, which we can actually put the naloxone in both males and females and knock out their copulatory preferences. It projects to the ventral tegmental area, which has dopamine neurons, that basically activate this whole CS-UCS integrator, which Pavlov didn't know existed, but we know exists thanks to the work of Kent Berridge, who studies drug addiction. This is what puts the CS and the UCS together. This is what puts the object together with your good feeling that makes the object then activate or prime your good feeling. So in closing, I think we really have sex for fun. And if we think about it, maybe all species on this planet have sex for pleasure most of the time. Mating can occur or not with a bit of luck in a mating context, but mating is not the critical variable. Pleasure is the critical variable. And I think desire for whom an individual mates with can work hand in hand with the desire for sex. That's kind of cool. And such sexual bonding, even if it's for a short time, requires conditioning and expectation to produce effective appetitive responses. So, Kraft-Ebing was right. First experiences matter. And I'll take any questions you might have. Am I good for time?
That's fine.
We're good? Okay. Thank you.